Hello and welcome. Today what I want to talk about is synapses. And synapses are a part of the nervous system where we find that neurons are able to communicate information between them. But before we talk about synapse, we have to remind ourselves about how information flows around in the nervous system. If we remember that a neuron has a bundle of fibres associated with it, and there are fundamentally two types of fibres associated with it. One set called dendrites and one called an axon. Now, dendrites have information flowing towards the cell body, and axons have information flowing away from the cell body. And just to remind ourselves that information flows as action potentials, which are short bursts of electrical activity, sort of like Morse code. So a sequence of action potentials is a information set, and this information flows down the dendrites towards the cell body and out the axon. Most neurons have many dendrites and a single axon. And the interesting part comes when that information needs to be passed on to the next neuron, and that's what we're going to talk about today. If we drew the next neuron sitting here like this, and there it is there, and there's a cell body there, and there's another axon heading out there, and let's just put a few of these dendrites in place. I'm just putting boxes on the end this time just to make it clear that that's the end of a dendrite. And just to remind ourselves, the information at the end of the day is going to flow from a dendrite towards the cell body and then out the axon. But what we are going to talk about today is what goes on here, where two neurons come close to each other and how that information is passed from one neuron to the next. Now, the most obvious and simple thing would be to electrically couple these two neurons. So in other words, as an action potential whizzes down here, the action potential could then just pass from one cell membrane to the next and out the other side into the next neuron. That process has some fundamental issues to talk about. If that was the case, there would be no way to have influence over the system. An influence really means decision making. If an action potential immediately went from one neuron to the next without missing a beat, without any change, the information transfer would be 100%. Now, in most cases that would be useful and fantastic, but what you want to be able to do is modulate information flow. In other words, some information you want to flow from one neuron to the next, but other information you would like to stop at this point. So you don't want the flow of action potentials to happen. So how that has evolved in the nervous system is to actually have, at this place called the synapse, a change in the way action potentials move. So we go from an electrical process where action potentials are differences in the ionic concentrations causing a potential difference between the inside of the neuron and the outside, and we change that mechanism of moving information to a chemical process. So in short, a synapse is a place where chemical transmission of information happens. And the reason for having this change in process is to allow us to influence whether information flows or not. So let's have a closer look at what a synapse looks like. Let's draw a very large scale here. Let's draw the end of the axon on this side, and let's draw the beginning of the new dendrite on this side. So this, we'll just put a D here for dendrite and an A for axon. Remember, info is flowing this way in this one and heading this way in this one. And the first and obvious thing to notice is that I've drawn a gap. There's a space. There is actually no contact, and so there is a gap here. And that gap, or cleft as it's sometimes called, is relatively small, and we are talking a gap of something like 90 nanometers. So it's a relatively small gap. So as the action potential comes down the axon, at the end of the axon, what we're going to do is we're going to change that action potential into a chemical messenger, and that chemical messenger is then going to move across towards the dendrite. So how does this happen? Well, the first important feature is at the end of an axon is a bundle of vesicles. And these vesicles have inside them neurotransmitter. And I'm just going to do those pink dots here. And neurotransmitter can be all sorts of molecules, mainly proteins, but it can be all sorts of different proteins. Different neurons use different neurotransmitters. And when an action potential comes along through a neuron, these vesicles are forced towards the cleft and they expel their neurotransmitter. So the neurotransmitter is expelled into the gap from these vesicles when an action potential comes along. And that neurotransmitter diffuses across the gap. And that's when we have to talk about the dendrite side. On the dendrite side are specific receptors sitting embedded in the cell membrane. And these receptors are highly specific for the particular neurotransmitter. So as the neurotransmitter comes across the gap, it attaches to these receptors. And these receptors then change their configuration, and what that means is that the arrangement, the three-dimensional arrangement of these proteins embedded in the cell membrane changes. And when that change happens, there is then an enzymatic chemical change, a chemical change within the dendrite side, and that causes the production of an action potential across the cell membrane that then allows the information to flow back down the dendrite. The last step to consider in this process is a step of recycling. This neurotransmitter is then released, and there are proteins in the membrane of the axon that reuptake the neurotransmitter and recycle it into vesicles back in the axon. And that will complete the cycle of an action potential moving through a synapse. Now do remember that this process is relatively slow. This process is slow in comparison to the movement of action potentials down nerve fibres. And this is what we call the rate limiting step. So in other words, when you want to do something very fast, say a reaction to a ball coming at you at a rapid rate, the principle of the nervous system will minimize the number of synapses in that process. Because it being the slowest part of the process, if you have less of it, information can travel more fast, and therefore you can get an action to happen rapidly. So this is the slow part. The process of an action potential across a synapse is the relatively slow part. So let's have a look at a diagram. In fact, this is from the Wikipedia page, so you can see this diagram here on Wikipedia, and please do have a look. And let's just run through it and check the elements that we can see. At the top of the page we have the axon, At the bottom of the page we have the dendrite, and here we have the vesicles, and within the vesicles we have the neurotransmitter. And what's going to happen is these vesicles, when an action potential comes along, are going to release their neurotransmitter into the gap, and there's the neurotransmitter being released into the gap. That's going to be taken up by the receptors on the dendrite side, that is then going to trigger a chemical reaction that will stimulate a new action potential in the dendrite, and then that neurotransmitter will then be recycled back into the axon. So here we can see that recycling process happening as well. So I hope you've got a clear picture now of how a synapse works in the nervous system. Thank you.